what commensal bacteria are is they're microbes that we have that are living all over our bodies, and they actually live also in our bodies. And for our research, we're particularly interested in those microbes that are inhabiting our gastrointestinal tract. And the reason for this is because within our gut, we actually have the largest population and density of bacteria in the world. We've got over 100 trillion different bacteria that are all residing and co-inhabiting the same niche. And so basically, what's really neat is these bacteria have co-evolved with us over time. And as a result, we've kind of grown and we have this mutualistic relationship with these bacteria and us as hosts. And what we get from them is things like these bacteria contribute to the development of our immune system. They help it to keep us healthy. They contribute to our metabolism by breaking down essential amino acids and vitamins that we can't do ourselves. And in fact, they can also protect us against pathogens. So at Manchester, we're interested in understanding how these bacteria contribute to our health. But also, maybe what's more interesting and more intriguing is we're interested in how these bacteria contribute to our health, but also when we introduce a pathogen or a parasite into that mix, how that then can alter how our health is and our overall well-being. And so we're interested in parasites like the whipworm, Trichuris, for instance. This is the model system that we're working on. And we're looking at the parasites, the microbiota, and the mice. And we're using a really specific model here at the University of Manchester. And that's using germ-free mice within our germ-free facility. And what we're able to do is we're able to breed and maintain both wild type and immune deficient mice within this facility. And these mice, germ-free mice, are completely devoid of all microorganisms. So they live quite happily in these HEPA-filtered isolators. They're given sterile water, sterile chow, sterile toys. And so that way we make sure that they are completely devoid of all bacteria. So then we can go back and we can then systematically add in different types of bacteria and then look at how a single species or multiple species of bacteria are then impacting our immune system and ultimately generating a healthy host. So what we can also do, which is very neat, is we can kind of look at this three-way relationship between the bacteria, the host, and the worm. But then we can start to kind of manipulate it, and we can use an immune deficient mouse and say, how does this deficiency in their adaptive immune system impact the bacteria or the worm? As well as in my research, what I'm doing is I'm looking at altering the bacteria themselves and looking at how bacteria that are missing an outer membrane protein are able to impact and influence the worm health and the host health. And even people are looking at doing molecular manipulation of the worm itself and how that then can influence this triad relationship. So why is this type of research important? Well, we're particularly interested in the whipworm Trichuris, and it's because it actually infects about over 600 million different people. And to date, we've got no effective means of controlling the infection. And it's a common human pathogen, and the one that we use in the lab, Trichuris mirus, is very closely related to the human worm, and it's a natural pathogen of mice. And so we know that the host immune system is really important for having parasitic infection. But also, what we've been able to decipher is the gut bacteria themselves are also really critical for whipworm infection. So what this means is we're actually able to identify different members or different components of the gut microbiota and the gut bacteria that are influencing how parasitic infections occur and what makes them successful. And by knowing this, it gives us an opportunity to then perhaps harness this as a potential microbial therapy in the future against not only Trichuris infections, but potentially other parasitic and other invading pathogens.